Good morning. Dr. Harbour and I have got it all worked out, and so I'm going to bring him back to stage. Thank you for your patience. Give him a warm welcome.
No, it's just a very clunky thing here. All right, can y'all hear me okay? Can somebody acknowledge?
We can hear you just fine, loud and clear.
Okay, thank you. All right, let's try this again. Modern technology, right? Let's try it again. We'll start with, we're going to talk about recent advances in uveal melanoma. And we're talking about the cycle of discovery that starts with patients. And we go to the laboratory to make discoveries that we then bring back to patients. And I mentioned the laboratory that I have, full of very talented young people. And these are some of the discoveries that we've made over the past few years, and I'll discuss some of these today. But the most important part of discovery are our patients, you, the patients who agree to participate in research. This allows us to understand better your own situation and also to make discoveries that will help other patients. So the cycle of discovery, it goes to the laboratory. We make discoveries in the laboratory, but then how do we bring it back to patients? Over a decade ago, I started the Collaborative Ocular Oncology Group to do just that. And the goal of this group, we call it the COOG, is to bring new discoveries from the laboratory to patients and to validate them through multi-center collaboration. So this is very different from retrospective research where you look back on time and look at different factors. We want to look prospectively, enrolling patients, trying new tests, trying new therapies. And most importantly, we want this to be based on science in uveal melanoma, not borrowing from other cancers, which has often been the case in the past. So in the first COOG study, this is where we actually validated the DecisionDx-UM test. Even to this day, almost a decade later, it's the only test that's been prospectively validated in a multi-center fashion. This test is now the most common by far used in the United States with a very high success rate. And we use this routinely now in the clinic to counsel patients about their risk of metastasis and to calibrate the amount of surveillance and the frequency of surveillance that they need to look for the development of metastatic disease. The COOG1 spurred further discovery. It led to the discovery of BAP1 mutations, which we reported in Science in 2010. We showed that this mutation is actually responsible for the class 2 gene expression profile, led to the discovery of the splicing factor SF3B1 in the class 1B tumors, and also to PRAME, which we'll talk about more. So now we're in the COOG2, the number 2 study from this group, and we just recently closed enrollment on this study. This is a 25-center study all across the United States and Canada, and here are the centers that participated in COOG2. And the goal here was to integrate the DecisionDx-UM, with the gene expression profile, with PRAME and with the NGS, which is next generation sequencing panel of all the mutations that are known to occur in uveal melanoma. And then also to incorporate this with chromosomal alterations. The CNVs refer to chromosomes. And our goal really is from a single needle biopsy, a safe, limited, small biopsy at the time the eye tumor is treated, that we can get all of this information that will help us to stratify patients for surveillance, to enroll high-risk patients into adjuvant or preventative therapy, and then to know what systemic therapies are likely to be effective in the future. Now, the latest direction for COOG is to expand beyond just ocular oncology into both ocular and medical oncology. So we're expanding to include medical oncologists across the United States and Canada, and we want to be able to enroll our high-risk patients, those that particularly have a class 2 gene expression profile into clinical trials of various preventative therapies, and then to develop better metastatic trials as well. This group is being led by Dr. Jose Lutsky here at University of Miami, collaborates with me, Dr. Rich Carvajal, and Dr. Takami Sato. We've already added about 15 additional medical oncologists around the country, and we're developing our clinical trials network. And what we're really focusing on is rational targets for uveal melanoma. In the past, because it was a rare disease, we really had to rely on pharmaceutical companies allowing us to use drugs that may have worked in a different cancer and hope that they might work in uveal melanoma, even though there wasn't really any rationale for it. But now that's all changing. We know very well the leading drivers of metastasis in uveal melanoma, and we can work directly with the pharmaceutical companies to identify targets that actually make sense. And the other important thing to mention here, it was not that long ago that uveal melanoma, being a rare cancer, that was a great hindrance in terms of getting pharmaceutical companies interested. Nowadays, the FDA has fast-tracked approval of drugs in the rare cancer setting like uveal melanoma. So pharmaceutical companies, particularly ones with new promising drugs, are really wanting to work with us to get a signal from their drug to get FDA approval. So things have really changed in the past few years, and we're having very fruitful collaborations with industry. So the three things we're going to focus on in the rest of this talk, these are the three things that we think are most important in driving metastasis in uveal melanoma. BAP1, which we already talked about. This is responsible for the class 2 gene expression profile. PRAME, which is abnormally expressed. It's not mutated, but it's expressed abnormally in some class 1s and in about half of class 2 tumors. And this further increases the risk of metastasis. And then finally, we're going to talk about LAG3, which you may not have heard about yet. So for BAP1, we've spent a lot of time and effort working on ways of therapeutically targeting BAP1. And for our purposes today, I'm just going to mention that we have identified a specific drug that is highly effective, at least in preclinical models. This is an HDAC4 inhibitor. Now, I'm sure many of you have heard about HDAC inhibitors, but there are many HDACs, and not all of them are relevant to uveal melanoma. We've now identified what we think is the most important HDAC, HDAC4, and we've identified a drug that hits this particular HDAC. And hopefully this will be published within the next week or two. And we're already talking to the company that has the license on this drug to start clinical trials in class 2 patients. And this would be, this is a well-tolerated pill that could be taken in the preventative setting to try to delay or prevent the development of metastatic disease. And then PRAME, as we mentioned, is a bad risk factor. It increases the risk of metastasis in both class 1 and class 2 patients. And we're understanding more and more about how to potentially target PRAME. The most promising avenue right now is immunotherapy. Because PRAME should not normally be expressed on adult cells, we can train the immune system to go after PRAME. And there are a number of companies working on this right now, and those in our COOG group are working with various pharmaceutical companies on potential clinical trials. I'm going to use the rest of this time to talk about checkpoint immunotherapy. And I'm sure many of you have heard about this, Keytruda or Pembro, Opdivo or Nivo, Yervoy, Ipi, or Ipi-Nivo. What all these refer to are checkpoint molecules that dampen down or turn off an immune response. And unfortunately, an inhibition of these molecules raises the immune response or increases the immune response in certain cancer types and allows your immune system to attack the cancer. This has been very successful in cutaneous metastatic melanoma, but unfortunately has not been very effective in uveal melanoma. And just before I try to explain why that might be, let's just take a quick look at what these molecules really do. You have something called T cells in your body that are immune cells that are involved in, for among other things, attacking a virus, let's say, that infects your body. And when the T cell sees this virus, it becomes activated. It has these molecules that become turned on that we indicate here in green. And this allows your body to fight that infection, or in some cases, cancers. And then at some point, once you resolve the infection, you really want to turn off those T cells so that they don't cause tissue destruction. And then you create memory cells. And then if you're ever exposed to that virus again, then you turn on the memory cells. This is what everybody's talking about right now with the coronavirus, that we need a vaccine to train our T cells to go after the coronavirus, and then to have memory cells that can come back if you're ever exposed to it again. But unfortunately, that doesn't always work well, and you end up with chronic infection or chronic inflammation, like with rheumatoid arthritis. And what happens here is that you never quite clear whatever is causing the immune response, the antigen that's causing the immune response. And so after a while, the body has to shut down these T cells or dampen them down so that they don't cause tissue destruction. And that's what we have indicated in red here. And this can happen in chronic inflammation, like in rheumatoid arthritis, and it can happen in cancer. And what are these molecules here? That is the CTLA-4 and the PD-1, what the Ipi-Nivo and these other compounds are trying to hit. It is trying to inhibit these things so that the T cells can be turned back on. But what we found is that LAG-3 is yet another one of these checkpoint molecules that may be more important in uveal melanoma and may explain why Ipi-Nivo and such compounds are not working that well. This is just an excerpt from our recent paper in Nature Communication, and I'll just draw your attention to the CTLA-4 and PD-1, where these little humps are very low here, meaning there's not much expression. But the LAG-3, you see these humps are much higher, meaning that most of the checkpoint marker expressed in uveal melanoma is LAG-3, not these other two. So based on this, we've already contracted with Bristol Myers Squibb, a big pharmaceutical company, to test their new LAG-3 inhibitor in patients with metastatic uveal melanoma. And if we see a signal here, then we want to rapidly expand this out into our COOG network, both in the metastatic setting and potentially in the adjuvant or preventative setting. So in summary, we're focusing our efforts on what we think are three of the most important targets that are responsible for most of the metastasis in uveal melanoma, which are BAP1, PRAME, and LAG-3. And we're using our collaborative ocular oncology network to reach as many patients as we can with potentially effective new therapies as soon as possible. I just want to finish with thanking our funding sources, Cure Insight, along with Cure-OM, Ocular Melanoma Foundation, and then National Cancer Institute, Melanoma Research Foundation, Melanoma Research Alliance. Without these very, very important sources of these private foundations, we could not keep this work going. You know, the National Cancer Institute gives out big grants, but it takes years to develop the data to get one of these grants. Whereas these private foundations, they allow us to move very quickly with new discoveries so that we can benefit patients more quickly. Thank you for your attention, and I'm happy to take any questions.
Thank you, Dr. Harbour. There was a lot of fantastic information, and I want to use this time to take questions if we can. All right, let's see here. I can pull up any that we have. If you have any questions, please feel free to drop them in the Q&A. We have one here from John. He says, can you give me your expected timeline for your HDAC4 project, and what would you expect the protocol for the trial to be?
It's a great question. We are working with the pharmaceutical company right now and have signed an agreement with them. And we are moving into the regulatory phase of getting the institutional review board agreement set up and so forth. I think realistically it could be 6 months or maybe even more. Unfortunately, some of these regulatory issues take a while, but usually the hardest step is actually getting the pharmaceutical company interested and in agreement to provide the drug. And this company is very, very interested. And we've already passed the most important hurdle. So, it's hard to put an exact timeline on it. Hopefully it'll be within a year. This drug has already been used in other settings, so we know that it's safe. So we don't have to go through a phase 1 safety trial. We can go right to an efficacy trial. And we would envision this first trial not being a randomized trial where some would get placebo, because patients at this stage, they don't want to be randomized to a placebo. But what we do have is over 1,000 patients with long follow-up from the COOG-1 and now the COOG-2 that we can use as a control comparison. So we can compare their outcomes to patients who get the drug to determine whether the drug is working. So we would envision enrolling patients in the adjuvant setting with class 2 uveal melanoma, and they would be assigned to take the drug. We would do certain blood tests and things like that to see how they're doing and how their body is reacting to the drug. And then, of course, the main outcome is to see if we can delay or prevent metastatic disease from developing.
Very good. Okay, thank you so much. That was from John, and that was his question. Thank you, John, for sending that in. Our next question is going to be, can you list your reference for your paper from Nature about LAG-3?
Yes, it's Nature Communication, and it was published this year in 2020. If you just Google my name and Nature Communication, it'll probably come up. You can just you should be able to find it.
Perfect. And if we find it too, we'll put it into your socio profile. And just so everyone knows, anything that's in socio is available for a year after this event. So recordings that are happening from this, links and information will be available to you if needed. I'm going to ask the next question. Is BAP1 always present in class 2?
BAP1 mutations are, we believe that they're virtually always present in class 2. Always, we try to avoid always and never because, nature doesn't follow those rules necessarily. But I would say the vast majority, over 90%, maybe 95% of class 2 tumors will have a BAP1 mutation. Now, we can't always find it because BAP1 mutations are, it's like finding a needle in a haystack. We have very good tools now, what we call next generation sequencing, that can find most of them, but some of them are very hard to find. But yes, virtually all class 2s will have a BAP1 mutation.
Okay, thank you. Okay, our next question comes from Andrea Spindler. Is there a known percentage risk factor for metastasis for those who are class 1A PRAME positive?
That is a very good question, and part of what we're answering in the COOG-2 trial is, which is, the class 1A and class 1B was based on a different algorithm that was embedded within the DecisionDx test, whereas PRAME was discovered as a completely new biomarker that was discovered independently of that. So we don't know how the 1A versus PRAME positivity will work out, but that's what we're studying right now. But I would say that the information that we have available right now, class 1A PRAME positive, the risk of metastasis, at least within the first 5 years, is going to be on the order of 15%, something like that. Still low, but not zero.
Okay, thank you so much, and everyone, have grace with me. I'm learning all kinds of new medical terms, and I'm pronouncing all of them as best as I can. But we have a few more questions. This is a very popular a session here, and so I want to go through some more of these, and we have plenty of time to answer about 5 or 6 more. I am type 2 PRAME positive, just diagnosed with numerous 7-centimeter spots in my liver. Can you recommend a trial for this presentation?
Well, I wouldn't want to. I can't give medical advice in this type of setting, but I would say that there are a number of trials, promising trials, that are already available, particularly in the immunotherapy realm. And you should discuss this with your medical oncologist, or if you reach out to me separately, I can put you in touch with a good medical oncologist who would be able to talk to you further. But there are several immunotherapy trials targeting some of the things that we just talked about here. And there are certain entry criteria. You have to have certain markers to be able to enter certain trials, and it gets a little complicated. But I would just say that there are several promising trials that you should be made aware of.
Thank you so much. And I've got and also you're going to be at your booth, virtual booth or lounge that's in the table view between this session and the next, and the session after that. So if you'd like to speak with him, he's got about 6 spots in his area if you'd like to visit him there. And then our next question is going to be, did you say that half 1A is PRAME positive?
No. So for class 1 tumors, about a third of them will be PRAME positive. And for class 2 tumors, about half of them will be PRAME positive.
Okay.
And so if you're class 1 and you're PRAME negative, you're class 1A and PRAME negative, your metastatic risk is under 5%. We're talking maybe 3%, something like that. If you have PRAME expression, that bumps up to maybe 15%, something like that. If you're class 2, the 5-year risk is about 70%, and then that bumps up to probably 80% if you're PRAME positive. So in both of them, it kind of makes things a little bit worse. But the hope is that PRAME can actually be a vulnerability, even though it makes the tumor more likely to spread. It might be a vulnerability that we can target therapeutically, and that's what I mentioned briefly in my talk.
Okay. Thank you for answering that. I'm going to get through these as quick as I can. Is there any work being done for conjunctival melanoma?
That's a great question, and I would just say real quickly that we often use the term ocular melanoma because people know what ocular means. A lot of people don't know what uveal means, but everybody here will be aware that there are actually 2 kinds of ocular melanoma. There's uveal melanoma, which is what we're talking about, and conjunctival melanoma, which is very different. Even though it's only a few millimeters away, it's completely different. And we've published on this showing that the mutations in conjunctival melanoma are completely different than uveal, but they're very similar to cutaneous melanoma. So there's growing evidence that you can treat conjunctival melanoma similarly to the way you treat cutaneous melanoma. For example, they often have BRAF mutations, which you don't see in uveal melanoma, but you see in cutaneous melanoma. And we have BRAF inhibitors. And there's also evidence that they are more immunoresponsive, like cutaneous melanomas, than are the uveal melanomas. We also see a UV signature, an ultraviolet light signature, with conjunctival melanoma, where we don't really see that for uveal melanoma, except for in the iris. The melanomas in the choroid and the ciliary body really don't get much UV light exposure, so we generally don't believe there's much association with ultraviolet light for the more posterior ones. But the conjunctiva obviously is very exposed to ultraviolet light. Now, why is that important? Well, ultraviolet light damage to DNA creates changes in the DNA that the immune system can sometimes recognize, what we call neoantigens. And that's one reason why cutaneous melanoma is so responsive to immunotherapy. And there's growing hope that will be the case with conjunctival melanoma as well.
Okay. All right, I have one from Colm Boren. He says, we had 2 biopsies and 2 different tests. Results were monostomy 3 and class 1A PRAME negative. What would those 2 disparate results indicate in terms of metastasis risk? I feel like I'm not pronouncing that correctly.
Metastasis risk, metastatic risk. Yeah. So there are some caveats because it depends on how the biopsy was done. Was it actually done in the same location? If one was done near the center of the tumor and the other was done somewhere else, or then the results could be different. But assuming that the 2 biopsies were done in a similar location, what we found in the COOG-1 study is that the class 1 versus class 2, the gene expression profile was usually correct, and the monosomy 3 was usually incorrect. So assuming that both biopsies were performed properly and in a good location, we would tend to rely on the class 1 gene expression profile.
Okay. Thank you very much. We have one from Susie. This one's a fun one. Just want to send a hi to Dr. Harbour for one of his 2005 plaque patients in St. Louis, and thanks for being our speaker today.
Oh, fantastic. That's great. Great to hear from you.
Yes. And then we have another one from John. We have, my diagnosis is 1A. I do not know positive or negative. How do I find that information?
Well, the PRAME was added to the test a few years ago. Castle will keep samples for a certain amount of time before they dispose of them. So some patients who were tested before PRAME became available may still have sample available that Castle could rerun for PRAME. So they could reach out and ask them about that. Just to reiterate something I said earlier, what all of these new things that we're adding, the mutations, the BAP1, all the other mutations that we're adding, all of this is we're doing this from a single needle biopsy, which no other test can really compare to. So we keep adding new things to it as they get validated. We don't really use them clinically until they've been validated. So the PRAME is now validated, and so they're using it clinically. But in the beginning, it was more of a research test. But you should reach out to Castle, and they may be able to either have the data that was only available in a research format, or they may be able to run it at this point.
Perfect. Thank you. And just so everyone knows, Castle Biosciences is one of our main sponsors of this event, and they have a sponsor profile that is in the Socio app if you'd like to know more information about them or get to their website quickly. And our next question comes from Diana. What is the percentage of MERS with class 1B PRAME positive and 1B PRAME negative?
So what's the percent of
MERS, MERS? That is the way it's written. Diana, if you don't mind putting in, METs. She says METs.
Oh, METs.
It was just a typo.
Okay, right. Yeah. So overall, roughly 75 or 80% of metastases will be class 2 with BAP1 mutations. Maybe about 15 to 20% will be class 1. And the vast majority of those will be class 1B PRAME positive. Okay. What we are learning is that the small percent of class 1As that metastasize mostly have PRAME expression. So that's what we're really sorting out. We're going to, when we are able to finish analyzing the COOG-2 data, we're going to come up with, we'll have these numbers very, very precisely determined. But at this point, I would say if it's, the percent of metastases that are class 1 will be about 15 or 20%. And the vast majority of those will be class 1B, PRAME positive.
Okay. All right. Thank you. So METS is the abbreviation for the word I'm having such a hard time with. So I'm going to use that for now. All right. We have another one from Colm. He said we had, we got this one here. We did this one. Let's see. Sherry, I'm in Canada and my biopsy showed monosomy 3, but no other information. Would that mean I have the BAP1 mutation and am class 2?
There is a lot of overlap. So, probably, if you look back at the Coog paper, it was published in Ophthalmology in 2012, and we can provide all these for you if there's a way to do that. I can give you the PDF so that people can get access.
Links, anything you have, send them all to me.
Okay. What that paper showed is that probably 75% of the time, class 2 was equivalent to monosomy 3. Okay, but in the 25% that they were discordant, where it was either class 1 monosomy 3 or class 2 disomy 3, that the vast majority of the time, the class 1, class 2 was correct in terms of predicting metastasis. So, there's at least I'd say there's a 75% chance, a good chance, that the monosomy 3 means that you are class 2. But it's just not as accurate. We're going for the highest accuracy that we can. But yes, if that's the data you have, then I would assume that you're class 2, and I would be doing surveillance accordingly.
Okay. All right, we have one from John. Do you expect the exclusion criteria for the HDAC4 trial to include any other adjuvant treatments that have already been tried?
That's a good question. I can't give you the answer to that at this time. We're still sort of discussing the entry criteria. I mean, certainly my preference would be not to exclude anyone, but we also have to think, what is the, in order to, what we're doing, these are called signal-seeking trials. So they're relatively small trials of 30 patients, give or take, and you're trying to get a signal to see if, some people do much better than you would have expected them to based on prior information. Okay. And then if you get that hit, that signal, then you open it up to multiple centers and you do a formal, bigger trial. So the best way to get a signal is you take the people at the very highest risk, and then you look for a reduction in their development of metastasis. So we're going to have to weigh those two factors. How do you get the greatest signal versus, have people been on therapy before? So I wish I could give you a definite answer, but my preference would certainly be not to exclude anyone if we can.
Okay. All right, our next question, we've got about 7 minutes. We're going to try and get through as many as we can. Do you collaborate with Will's Eye, or are you both talking different tracks to treatment?
I would say that the medical oncology group that collaborates with Will's Eye, we're very much working with them. Dr. Takami Sato is on our leadership board of the Coog. He's there at Thomas Jefferson in Philadelphia. And our research, I actually have a Teen Science Award from the Melanoma Research Foundation working with the Thomas Jefferson group. So in the research area, we work very closely with them and have very full collaborations with them.
Okay. All right. And then we have our next question, which has been voted up a couple of times. Are you looking for more class 2 PRAME positive patients for your drug testing clinical trials?
Yes, this would be the group that we're most interested in for the adjuvant or preventative trials, because we know that these are patients that are in a high-risk group, and that we want to help them, and also would be most likely to be able to see a signal if the drug is working in that group. So yes.
Okay, and how do they apply to do that? What's the process of doing that?
For now, you can reach out to me, and I'll provide my contact information. I did have a website that I didn't mention by name, but it was on the on my slide presentation. It's coog.life, COOG.life, and we're still it's still being built out, so the information is not there yet. But as soon as we have trials available or that are coming online, we will announce them through this website.
Perfect. And I know that my team has probably put that website link into the chat because they're wonderful. Okay, thank you for that question. And I think you might have covered this earlier, but Trish might not have been here. Are 1/2 of 1A PRAME positive? Are 1/2 of 1A PRAME or 1/2 of 1A PRAME positive?
Yeah, I think I answered that before. That about a third of all class 1 patients will be PRAME positive, but most of those will be 1B. There are some 1As that are PRAME positive too, and that's what we're trying to sort out. It's what how do we sort out when they're 1A PRAME positive versus 1A PRAME negative? But certainly, if they're 1A PRAME negative, this is the lowest risk group.
Okay.
But when they're 1A, which is supposed to be a low risk biomarker, and PRAME positive, which is the more important, just like we showed that between monosomy 3 and class 2, most of the time they overlap, but when they disagreed, usually it was the class 2 that was more accurate. We'll have to figure that out between the PRAME and the 1A, 1B, we're very close to doing that now.
Okay, perfect. If a patient was unable to have biopsy, what will be available to predict Mets?
Yeah, well, this is what we're actively working on now, and this is liquid biopsy. And what this means is taking something from the blood that we can analyze and look for circulating biomarkers in the blood. That might be a protein, it might be a piece of DNA that we sequence, but that's really the next phase of COOG, what will be COOG 3, is to look for biomarkers that we don't require biopsy, but we can assay that from the blood.
Okay, that's fascinating. I've got here, if you do not have tumor tissue due to radiation, are there any peripheral blood tests, genetic marker tests for Mets choroid melanoma patients?
Yeah, that's the same answer to the previous question. The answer is yes. We're working with multiple different angles on this. There are different types of circulating biomarkers. There can be cell-free DNA, just meaning turns out that we have little pieces of DNA floating around in our blood. And some of those may be telling us that you're class 2 or you're class 1. There are things called exosomes, which are little tiny vesicles full of cargo that are protein, DNA, RNA, and those may actually be able to tell us, that somebody is class 1 or class 2. And then there are other methods, and we're looking at all of those methods in parallel to see if any of these would be effective, both in terms of patients who weren't able to get a biopsy, but also in terms of monitoring patients. Let's say a patient's at high risk and they're class 2, and we monitor their blood and we look for a marker and it's low, low, low. And then when that marker starts going up, then we know that we may need to look more closely for potential early metastasis. So there are a lot of ways that we can use these blood circulating biomarkers.
Okay. I've got about 2 minutes left, so I want to rapid fire these questions because I want to get everybody's in. Lynn says, had class 1A PRAME from August 2019, 10 by 10 by 5.7. How often should I be biopsied? Been followed for 18 years at UNC Kittner Eye Center.
Well, the profile that you just gave would be a very low risk lesion. We generally don't believe that tumor size and physical features of the tumor are very helpful because all of our measurements are a little different. So one person's 10 millimeters is another person's 11 millimeters. But we did find, and this has been validated by several other groups, that there's something kind of special about 12 millimeter diameter. We don't know why, but if the diameter is under 12 millimeters, the risk of metastasis goes way down. And that's probably because the tumor hasn't been there long enough to kind of learn how to escape. That's kind of the way I think about it. So that 10 millimeter diameter plus the relatively the good genetic profile, that would mean that the risk is really pretty low. For my patients like that, we generally get an ultrasound of the liver once a year and a chest x-ray once a year, something like that. And then you have to decide after 7 to 10 years, do you really need to keep going? And we have a discussion with each patient individually and decide, because the science, the data, the evidence starts to get very thin after you get out to 7 to 10 years. What's the value of continuing to do that?
Okay. So we're at our time here, but I have 2 more questions. I just want you to simply a yes or no for each of these. And if there's more that you'd like to expand or they'd like to talk about, they can meet you in your lounge. The first is, do you recommend any surveillance or scans after 10 years? I'm almost 11 years out from treatment for a small choroidal melanoma and hoping I'm out of the woods as far as choroidal melanoma at this point.
I just answered it with the last one, and there's not strong data either way. So the value is certainly very low at 10 years out of continuing the surveillance.
Okay. And then our last one is, would class 2 PRAME negative qualify for the trial for adjuvant therapy that you just mentioned?
Most likely, yes. Most likely, yes.
Okay. Well, thank you so much for your time. There was lots of questions, and it was a really valuable session. And again, if you want to hear more from Dr. Harbour, we're going to put some information into his speaker profile. And also we're going to put this information, we'll have a recording that's ready, available tomorrow for everyone to view if you missed the first part of that. And we appreciate your time. Thank you so much.
Thank you, Bree, and thanks everyone. I apologize for my technical challenges there at the beginning, but thank you all very much.
You can do this. All right, you have a great afternoon, okay?
Take care.
All right, everyone, we're going to go to a short break, and we'll be right back.